Well, first I want to say thanks to Scott and Stacy and all the people here who asked me to participate as the keynote. This is a milestone. I've done this at other places, but I never made it up to Boston. So my son will be proud of me. But anyway, I also want to thank Danielle Camp for the tremendous work that she has done in putting this together and supporting all the effort. I'm going to begin with the first slide, please.
You have a little clicker right there.
This one?
Yep. Right button.
Right button. There you go. You know, so what happened when I got to ATCC is I had to change my purely, research career, research program career into one that combined business and science together. One of the techniques that we use in management and management sciences is case studies. Okay, it's interesting. We're in the Sloan School where they do those kinds of things all the time. So I'm going to start off and talk about 3 case studies for you and the lessons learned from those case studies. So the first thing we find out is there was an interesting scientist who in the late 50s and then in the 60s and 70s, his name was Walter Nelson Rees, decided that he wanted to look at the state of affairs in cell systems, cell cultures. He was a member of the Naval Laboratory in California, and he began to do validating cell line, work, and he found out that at least 30 or 40% of the cell lines that he was looking at were invalid. He was doing karyotyping analysis, and then he did something that was strategically a disaster. He published a series of papers in Science highlighting the mistakes in publications by the field. And he quickly became a pariah, and his voice was silenced, and we didn't hear much about him, except in his studies, he was very, very early in demonstrating the tremendous contamination of cell lines by HeLa cells. And there's been a great deal of discussion about, and as you know, it's reached a high point recently with the story with Henrietta Lacks. So that's a negative one, and the negative is that if no matter what the value of the work that you've done, if you don't communicate it well, if you don't address it with a proper strategy, it's going to be reversed and it's going to become a negative. Okay, so the next study I want to talk about is the Bacteriological Code Revised. This is 1975, and I have the key paper here that came out of it. This is International Journal of Systematics and Evolutionary Microbiology, valid publication of names of prokaryotes according to the rules of nomenclature, past history, and current practice. Well, this is a success story. And the success story was that they developed in this approach a system of valid publication in several journals, a system of prokaryotic nomenclature, which is a desperate need in all the fields we've been talking about, a governance structure, and we're going to repeat that over and over, a global governance structure, so to speak, and a system for storing materials standards in they weren't called biological resource centers, they were just collections or repositories. Interestingly enough, they named ATCC as the principal site for that storage. At that time, there was not that many repositories. Now there's 481 repositories, very few biological resource centers, and now materials are deposited all over the world, and you're required to deposit it in 2 places, and you need to have a publication, and you must share the material. So those are the elements of that success story of a governance, a system of publication, of communication, of validation of materials and culture collections, and the sharing of the material. So I think that's a lessons learned. And then we go to the next study, and that's the Budapest Treaty for Patents. And the Budapest Treaty for Patents is, again, a system of governance, and it's a global system for storing and exchanging patent material. You know, you're required to put a prototype biological material if you want to patent it. And they created organizations to receive that material, international depository authorities. There were 5 at that time, and ATCC was one of them. And system for regulatory coordination and compliance, and it's a great success. It brought law and order, and it brought a framework for working with patents in the biological system. At this point, ATCC holds 35,000 patents from that program. It's the largest. We have about 58% of the world's patents, many of them which are off patent. That's another interesting point. Okay, so in 1993, in the middle of 1993, I'm doing my job as a vice provost and graduate dean, and one of my former colleagues, Julie Youngder at Pittsburgh, my chairman at Pittsburgh, calls me up and he says, Ray, you've got to go look at ATCC. There's really, really problems there, and we need your help. And the search committee was very interesting. Bartharella was on that search committee. Elliot Levine from the Wistar was on that search committee. And I came in, and they asked me the typical question that you ask for leadership people. What's your vision for ATCC? Okay. And then I surprised them. And I said, my vision for ATCC is 2 things. That it's going to be the standards organization, that it is the standards organization, and that's its future as a standards organization. And the second thing I said to them was that ATCC is an information and knowledge organization, and you have to build around that idea as well, okay? That's 1993. I'm there for a short period of time, and I meet an individual named Lois Blaine, okay? Lois Blaine. And Lois has a very interesting title. Her title is Director of Bioinformatics. Remember, this is 1993, okay? It hadn't become fashionable yet. It was just beginning to emerge. She was Director of Bioinformatics, and her field was the standardization of information, and she had a very good background from IBM and the National Library of Medicine. And we got together, and we started talking, and because of her discussions with me, I changed the mission statement of ATCC to the following: to acquire, authenticate, preserve, develop, standardize, and distribute biological materials and information. So I linked the idea of biomaterials and information together and said, you can't separate them. They're one and the same. And you have to focus on the issue of information for the advancement and application of scientific knowledge. Then I did a second thing. I created a DNA for the ATCC, and that DNA was APAD, okay, as you could see. And instead of having nucleotides in a typical DNA, these were resource units or resource centers within ATCC, which sequenced, produced a standards process, and at the end of it, you had an outcome of materials that were going to be distributed. Now, I put authentication and development in red for a particular reason, because the key to being a biological resource center and not just a collection is authentication and development, okay? There are many collections, repositories, but they don't do authentication the way we do authentication, and I was going to put a lot of emphasis on authentication. We have an authentication lab at ATCC, which needs further development. But the second key point was the word development. And it was my belief that you don't get any respect in science unless you do R&D, okay? So if ATCC wanted to be respected as a scientific organization and not just a biobank, okay, or a utility, it had to do research. So then the question is, what kind of research do you do at ATCC? Well, that's a whole other story. But one of the things you do is standardization research, okay? And I'm going to get back to that as a recommendation. Okay. So let's go back to the milestones, again, in the evolution of biological standards. And in about 1999, after a series of meetings, I made a great discovery after I was at ATCC. I found out that Joshua Lederberg, Dr. Joshua Lederberg, was on my board, but had been inactive. And I went to New York to see Dr. Lederberg. And I got to say, some of the most enjoyable highlights of my career at ATCC was the 3 or 4 trips to New York. and visiting with Dr. Lederberg in his stacks of papers and materials, and occasionally having lunch with him in the Rockefeller cafeteria. And he said two very interesting things to me. He said many, many things to me. Brilliant, brilliant man. And he said, first, that ATCC and biological resource centers were essential to the scientific enterprise. Well, he was preaching to the converted already. That wasn't a big problem. But then he said a second thing. He said, in order to have impact, in order for biological resource centers to have impact, you had to prove the economic value of that resource, okay? And you had to prove it in Washington, where the people were who gave out money for such a thing. Okay, so that set me off in my neighborhood, so to speak, and I found out, and one of my social contacts was a guy named Bob Lighton. And Bob Lighton was the head of economics at the Brookings Institute. I went to Bob, I told him about the idea, and he said, great idea, let's go do it. He says, I know 3 or 4 people who could probably do this, but he says, let's try someone named Scott Stern. He happens to be a fellow at the Brookings Institute. And I said, great, and I went to see Scott, and he jumped on it right away. He was a young assistant professor. He was a research fellow at the National Bureau of Economic Research, remember Scott? Okay. And assistant professor of management. And he was a fellow. And we got together, and I provided him a lot of input. And from that collaboration came out the Biological Resource Centers, the Blue Book, as we like to call it, okay? And he had a lot of help. Is Jeff here? Is Jeff Furman here? Okay, so Jeff Furman was his graduate student, who's now a faculty member at BU. And they teamed up and did what they should have done, put out a series of scholarly articles and presentations and a book. And there's one, I'm going to just cite chapter 3, the title of chapter 3. You ought to read this book, it's very good. Climbing Atop the Shoulders of Giants, BRCs as Cumulative Knowledge Hubs. And I loved it because he hit at that whole idea of information and knowledge as part of the BRC. So, while he was doing that, and I don't think he knew I was doing this, I put together this book called Biological Resource Centers. And I did it this way so that you'd think it was just easy read, a small, thin little book, but, it's a big book. And it had 15 authors in it, the leading people in the field. And it's Biological Resource Centers, their impact on the scientific community and the global economy, okay? And in this book, and I think there's some very critical articles here, is the article by Lois Blaine, okay? And Lois writes, knowledge management and bioinformatics. She brings home again the point of what the role of a biological resource center is. And another chapter in this book is by Frank Simeone. Frank, you're here, aren't you? 39 years employee of ATCC, Mr. ATCC. I know about half of what he knows, okay? Regulatory coordination and compliance, okay? We're going to be dealing with that if we're going to be dealing with standards. And then we go to chapter 17 by a guy named Scott Stern, and it's called An Economic Perspective. And so he reinforces what he did in the Little Blue Book. Now, I'm going to save the conclusion because there's some recommendations I made in the conclusion that I'm going to get back to, okay? So thanks for bearing with me on this part of the story. And then about 2007, I have argued all along that you have to be recognized and certified as a standards organization. So I teamed up with ANSI, and ATCC became a standards development organization. And we took on the task of making 2 standards right away. The first standard was an anthrax toxin. I mean, this was very, very topical at the time. And then we took on the task of making a cell standard, okay? And the cell standard was the ATC cell authentication standard, the standard put together by ANSI and ATCC for human cell lines. John Masters probably talked about it this morning, I'm sure, because John was the co-leader of the consensus group. Is Yvonne here? Yvonne? Yvonne was the co-chair, one of our employees, and she's gone around the country, talking up this issue, describes optical cell line authentication. And I asked Yvonne to look at this topic early on because I felt that the cell authentication would have a great impact on the field. Again, I'm going to talk about impact later. You have to look at impact if you want to try to make some progress in a field like as wide as this is. And after ATC standard, multiple journals require cell line authentication, and we'll hear more about that later. Okay. We've been talking about that all day today. Do standards matter? And I think we would have to say, yes, they do matter. And the question is, to answer a question whether something matters, you have to do a landscape analysis. Now, we do that in business all the time, okay? Situational analysis, landscape analysis. We do that in science. We do that in a lot of things. So the question is current standards, landscape, and life science. And the framework is, it's one of my favorites, is why, what, how, and who, okay? And that's how you approach the question of landscaping, okay? And so let's talk about why. Now, we've been talking about why pretty much all morning long, and so I'm not going to go over it in great detail, but I think this slide really, really hits the question that we've got complexity issues because of the changes in science and what we're doing and how we're doing it. We got the issue of competition, we got the issue of community, and we have the issue of language, okay? And I think the issue of language is very significant in whether we're going to make progress or not going to make progress. And why? Because we have increasing complexity and collaboration in the way we do science today. You know, the individual scientist with a small R01, like the way I began, 100,000, okay? Now, you know, you probably couldn't pay for, you know, half of your salary and the supplies. So collaborative environment decreases individual control over experiments. Okay, it's a team sport, and you have to really work at keeping the team on top of what. You know, there are lab directors, and I know, who still meet with their postdocs and their graduate students every week, and they go over data, but that's not really the usual thing anymore. They don't have time for that anymore. They're busy writing grants. Why? There's a lack of standards in the life science research. There's lack of standards in material standards, as well as written consensus standards, and I also would say in process standards. An example of a material sample is HERC32 cell line. Example of a written consensus of laboratory animal welfare. So the conclusion, by looking at the effect of these standards, is that standards improve life science research quality, but few life science research standards exist. I think we'll all agree after today that that's a truth. So why? The irreproducibility in life science research. Now, I'm not going to go through the rest of this because we've been talking about this all morning, but irreproducibility of research findings affects all stakeholders. It's a domino effect. And this is the hardest thing to explain to people, that the mistakes you make early on in the process in basic research will have an effect on translational research, will have an effect on clinical research, and you have to have this continuum of standards. Well, what's another why? Another why, and this is some data we've put together, it's going to be part of this publication from GBSI, is that economic challenges and competition decrease resources available for each publication, okay? And it's going to get worse before it gets better, but that's pretty solid data. Why? Misidentified cell lines. Fewer than 50% of researchers regularly verify the identity of their cell lines. Many errors are unrecognized. So I'll give you data from ATCC. This is over 30, 40 years. We see 30% misidentified materials coming into ATCC. Okay, and materials are not coming into ATCC, and I'm going to get back to that in a moment. So they're ubiquitous in labs and literature. And the question, why are they so ubiquitous? Because they're not on the scientist's to-do list. Okay, when they go through all the things you need to do for a publication or you want to do a research program, it's not on that list, okay? You go down the hall and you borrow a cell line from somebody who's working in the field. You don't check it for mycoplasma. You don't check it for contamination, adventitious agents. You just borrow it because you trust it it's going to be good. You don't know the passages of it, okay? You don't have any idea of its traceability, okay? But you borrow it like a cup of sugar when you're baking, and that's it. And most of the young investigators really don't have much training and experience in cell culture, okay? And so we've lost that art, okay? Everybody's into kits. You want to do something, buy this kit, buy that kit. That's the way you do things. So I'm going to skip this because you've heard about it, and I want to go to the what. So how are we going to solve this problem? Well, these are my recommendations. We need an enhanced governance system, something similar to what we did with patent deposits, something what we did with type cultures. We need a harmonized consensus-based life science standards. The key word there is harmonized because we're dealing with a global scientific community. We need dictionary and working language. There's no field of science, and you could study different fields that have made progress until they've come up with a dictionary of terms and everybody worked with those terms. And then you had a communication that developed that was workable. You've got to have a dictionary of terms and a working language. The quality control at critical points in the R&D process, you got to know where you want to make your insertions, where you want to do your QC. You need harmonization of regulatory agency policies and practices. You want industry to stand up and tell you about that? Okay. They'll tell you the nightmare trying to deal with that. You need supportive journal policies, and I'm very pleased to see what's happening in that area. I think they're critical to the success of what we're talking about. And you need education and training. How many graduate students get any training in the area of QC or anything to do with cell culture or the things we're talking about? So you need optimization of research reproducibility. Now, in order for us to make progress in this field, okay, we're going to have to engage a large group of stakeholders. And I've just given you a map of the critical individuals, institutions, and programs that are going to have to be integrated to work. So standards development requires engagement of many diverse and siloed stakeholders, because there's a lot of silos in the community that we work in. And there's many silos in single departments or in institutions, so you can imagine what it is on a global basis. And I think the thing we have to do to move forward is to emphasize what I call the 4 C's: communication, content, culture, and community, okay? And I think this has to be a focus of our attention in trying to go forward. And I want to say something about culture because it's not always as appreciated as it should, but we're going to have to bring the social scientists into some of this work that we're talking about, okay? Not only the economists. You still think yourself a social scientist, this guy? Oh, okay. But we're going to have to bring in the sociologists, the anthropologists. Because guess what? We're dealing with behavior. A lot of what we're talking about is behavior. I mean, for example, we know that 8 of the 10 most important chronic diseases in the world are all related to behavior issues. Okay? It's the same thing in this problem here. A lot of what we're talking about is behavior. But it's not just behavior. We got challenges of measurement. So now, what's my recommendation at this point of what I think are the ways we can go forward and where we're going to get the most return? I'm going to borrow a comment that was made to me by one of my colleagues, Matt Klusas, who's in the audience. Matt loves this comment. I love it as much as he does. When you have a problem like this, don't boil the ocean. Okay? Don't boil the ocean. This is an ocean we're talking about out there in the standards field in the life sciences. You can't possibly try to do anything by taking it all on at the same time. So you have to have prioritization. You have to have focus. You have to have analysis of impact factors on what will you do that brings you a return on investment. It's like any other business kind of thing. The ROI is very important. What's going to cost you to do this? What are you going to get back out of it? What's the return going to be? And I think that one of the critical determinants of success or progress in this field is going to be the ease of measurement. This is for Bill Bentley, my colleague here from Maryland. The ease of measurement, the metrology. NIST is all about metrology. Which are the things that are going to lend themselves to measurement? Those are the ones that you want to play with first, second, and third. So, all right, Ray, what would you do? Where would you go if you were given this problem? What are the areas that you would go? So I'm going to give you, for whatever it's worth, where I think to go. I would work very, very hard on information standards. It lends itself to what we're trying to do, okay? The field of IT is so far ahead of everything that we could do something with that field very, very rapidly. On the biology side, I think antibodies. And I'm just not thinking about it. I'm going to do something about it. And I think antibodies because it transcends basic, translational, and clinical research. And I think it's very, very important, as we know, in clinical therapy today. And it makes the most sense that we could do something with antibodies. And we're going to start something. Where else would I go? I would go synthetic biology. And I'll tell you why, because I think synthetic biology is measurable. And I'm very proud to tell you, go back to what I mentioned about D, the part of the research. ATCC has offered 2 synthetic biology reference standards in the last 6 months. One for West Nile virus and one for norovirus. Now, the norovirus one is particularly interesting because you can't culture it very easily. And synthetic biology allowed us to break through that barrier and put out a reference standard. So I think synthetic biology, and you know, it's just coming on, gangbusters. And then finally, I've been saying this for a long time, I think we ought to look at, Laurie Borders in the audience, so she knows I'm going to say this. I think we got one of my board members from MIT. Consent and procurement. Consent and procurement of tissues. It's easy. It's doable. We're doing it all the time in different places around the country, but we didn't harmonize it. We haven't harmonized it in the country or globally. We can do consent and procurement and what follows. So with that, I'm going to just say that one of the ways that I looked at how to get this done was to create an institute, a nonprofit institute whose mission was thought leadership, policy, and training. And I suggested the development of the GBSI, and my board agreed with me. And we were able to fund that institute about 2 years ago, and the rest you're hearing about today. So I think, to me, this is a critical infrastructure that is necessary to partner with the various elements in the scientific community to move this field forward. Thank you for your time and attention.
A few minutes maybe for questions, and before we do kind of go to our deep dives here.
So what do you mean about antibodies, going for antibodies?
What I have been told by a number of people is the field is full of junk. That you can buy antibodies from many places, and a lot of it is not very good. And you can go back to that same place, and you'll still get junk. So we need some kind of a filtering system. We need some kind of a quality control system on antibodies. I think 20 years ago, antibodies was, we were talking about use in clinical therapy. 20 years later, we're doing it. Maybe iPS cells and stem cells do that 20 years from now, right, Laurie? But I think antibodies lend themselves to what we're trying to do, and I think we can clean up that and make significant progress. Are we going to totally clean it up? No. But I think we can make very significant progress, Joanne, in antibodies. Yeah.
Who do you see as the key stakeholders necessary to make GBSI successful? Who has to be at the table and signed up?
Well, I'm prejudiced. To me, always I would say the scientists, the people doing the work in science, okay, and putting out the output of that. I think, though, I think the journals have a tremendous role to play in this. I think the journals are a critical gatekeeper in the process, and they could influence the quality of the materials and the validity of the work that's being done. I think the agencies can do the same thing in parallel with the journals. I think the submission of grants should have a certain requirement for standardization or, of documentation of standardization of materials. I think, as all things today in the information age, I think an advocacy campaign, a really well-designed advocacy campaign to explain to the various stakeholders why this is important and what the outcome would be for everybody, the success that will come out of it is very, very important. So I think these are all critical elements in what we have to do. I would recommend one other thing, and I mentioned it today. I think we need a stimulus for serious research on standards. There has to be a research program underpinning standards, or it's never going to get the respect of the scientific community. And I think there should be, and we talked about it at lunch, either there is a section in the current journals where there is attention paid to the standards, rather, the other alternative, you start a journal. Everybody wants to start a journal for standards. No, I don't like that. I'd rather see it as part of the system we're in right now. But I think the journal should put attention to that. They may have a section in their journal that's run by a sub-editor that deals with standards and specialists in the field, and that would be very, very good. And I think this issue of errata and publishing of errata, I want to bring out one other point, and that is a lot of this problem came about because of the change in the way we dealt with intellectual property in biology. Okay? I'll give you facts behind it. After 1983, ATCC was not one biological material was donated to ATCC that had any serious impact on the field. Okay? What happened was, with the change in attitude about intellectual property, the laws, the Bayh-Dole, and the patenting of biological materials, the growth of technology transfer offices, vice presidents for research and all that, you lost a QC. The QC step was taken out of the system because when you sent the material into the collections, they did QC to determine whether it was valid or not. Well, that was now removed. You didn't have to do it. And the QC is not taking place in the universities within the structure that they're working with. And so I argue very, very much that that has been a two-edged sword. Yes, it stimulated a lot of biotechnology development, but it increased the problem of irreducibility of research. I was going to tell you that. I just remembered I had to tell you that. Any other questions?
I think, well, listen, thank you very, very much. And I think really, very, very